Welcome to the virtual lecture on Chapter 3, Part 2, The Molecules of Cells. In this video, we're going to cover 3 of the 4 major classes of biomolecules, those classes being the carbohydrates, lipids, and nucleic acids. The first class we're going to cover are the carbohydrates. And if you remember from the Part 1 video, the monomers, the building block molecules of this class, are called monosaccharides. Another name for a monosaccharide is a simple sugar. And if you put 2 or more monosaccharides together and you link them together into a chain, you then have a polymer, which is referred to as a polysaccharide. In other words, a complex sugar. So instead of giving the entire class of carbohydrates a one-sentence description of their function, we're going to divide this class into monosaccharides first, and then we'll talk about the polysaccharides second. So starting with monosaccharides, their basic function is to supply energy for cells or organisms that consume them. So monosaccharides are an immediate source of energy. We will devote an entire chapter on a process called cellular respiration, which is all about how an individual cell takes a monosaccharide like glucose and completely breaks down this molecule into carbon dioxide, water, and in the process releases quite a bit of energy to power the cell, to power the whole organism. So as I have said here at the bottom, when a cell breaks down a monosaccharide, it releases energy that cell can then use for itself. Also, organisms, if they have an excess of monosaccharides, like for instance, if you have a high level of glucose in your blood, we have the ability, or many organisms have the ability, to store excess monosaccharides in the short term so they can use it at a later point in time. Both plants and animals can do that. They do so in a slightly different way. So the most important monosaccharide is glucose. I call it the universal cellular fuel because pretty much every living thing, every cell, prefers glucose as an energy source. So it's commonly broken down to provide energy. So for instance, if you look at your body, your brain burns, a pound of glucose in a day. Your muscle fiber cells use a tremendous amount of glucose. So even if you're not consuming glucose itself, many other foods are, at some point or another, converted into glucose when they are broken down. There are other major biologically relevant monosaccharides. I'm going to cover 2. Those are fructose, also known as fruit sugar. And as the name implies, it's commonly found in fruits, also in honey, some vegetables as well, underground. And the other major monosaccharide is called galactose. And that is basically half of a disaccharide sugar, which we're going to learn about, called lactose. So it's a component of milk sugar. So if an animal, like yourself, has the enzyme that allows you to digest lactose in milk, then when you digest that lactose, you're left with 2 sugars. Those are glucose and galactose. Importantly, both of these monosaccharides, fructose and galactose, when you break them down, they are converted into glucose. So what I'm saying is basically it doesn't matter if you're consuming glucose, fructose, or galactose, they all result in glucose, and they all result in the same amount of energy. So, in other words, we metabolize these three monosaccharides in the same manner. And since glucose is so important, it's this universal cellular fuel that powers life, it's one of the few molecules that I want you to be comfortable recognizing in terms of its basic structure. So here's glucose on the right. And since it's important, we're going to jump over to my drawing, and we're going to draw it out again on our own. So first off, I want to point out glucose. You know, for instance, like water. Water is a simple molecule. It's H2O. There's 2 hydrogens for every 1 oxygen. Glucose, its formula is a little bit more complex. There's 6 carbons, 12 hydrogens, and 6 oxygens. That's where it gets the name carbon, hydrate for water. There's 1 water molecule for every carbon atom. So you can kind of think of it that way. Carbohydrate. Now, I've started the drawing of glucose, but you need to realize that, because I understand you don't have a very in-depth chemistry background, which is fine, but we're using a shorthand depiction of a complex molecule, in this case glucose. So what's going on here? Well, if you look at this shape, it has, in a sense, like 6 corners to it. Now, each corner is a shorthand representation of a carbon atom. So I want to put a dot for each carbon atom. So there's 1, 2, 3, 4, 5, and then number 6 is up here at the top, hanging off of this ring. So that's glucose. And usually we number the carbons, starting with this one here and moving in a clockwise direction. So this is number 1, this is number 2, 3, 4, 5, and then 6 up there. The only other part that I left out is the hydrogens. So let me fill that in. And think about it. If each of those dots represents a carbon atom, at this point in the class, you should know that carbon, because of its unique valence structure, always likes to form 4 covalent bonds. And currently, if we look at carbon number 1 right here, that carbon's not so happy, right? It only has 3 lines coming from it, so it needs 1 more bond. So that's where that hydrogen's going to come in. So there's a hydrogen there, here, and that's it. If you were to add up all those carbons, hydrogens, and oxygens, it would come out to C6H12O6. That's glucose. So you definitely do not need to know how to draw this molecule from memory, but be able to recognize it. So for instance, if I give you a variety of important molecules like a nucleotide, an amino acid, a fat, a phospholipid, and glucose, be able to know, okay, well, that's glucose, but it's obviously not a fat. It's obviously not, one of those other molecules. So be able to distinguish and recognize it. One last thing I want to point out about glucose, which relates to our first part 1 video from chapter 3. Remember I said one of those 4 functional groups that we covered, hydroxyl groups, right? OH. I told you in the part 1 video that hydroxyl groups are commonly found in carbohydrates, which glucose is an example of. And you can see that right here. There's how many? 1, 2, 3, 4, 5 of them. Okay? And that's why glucose loves water. Because for each of these hydroxyl groups, I have to draw it really small here, but there's a negative oxygen and a positive hydrogen. So that's responsible for this molecule's ability to form hydrogen bonds, and it's responsible for it being hydrophilic. moving forward, if you put two monosaccharides together into like a chain of two, we don't consider that a polysaccharide. To be a polysaccharide, you need three or more. So we have a special name for when you have two monosaccharides covalently attached to one another. We call that a disaccharide, di for two. And there are three biologically relevant disaccharides that I want you to be familiar with. Those are lactose, which is milk sugar. And you should know that lactose is composed of glucose attached to galactose. Second is sucrose, which is commonly just known as sugar. Okay, that's table sugar from sugarcane. And sucrose is made up of glucose bound to fructose. And then lastly, maltose, known as grain sugar, is kind of a precursor to starch, and it's composed of glucose bound to another glucose. So if you take any of these three disaccharides, think about what would happen to them in your digestive system, right? In your stomach, in your small intestine, even in your spit. You have enzymes that are going to, in your digestive system, break these bonds, thereby releasing the individual monosaccharides. Then your small intestine is going to absorb these monosaccharides. It's going to go into your bloodstream. It will then be sent to your liver to be processed. Depending on the metabolic needs of your body at that current point in time, many of these monosaccharides may be sent to any of the cells from your brain down to your toes that need energy. And as we learned a few moments ago, you would metabolize and you would break down all of these disaccharides really in the same way, because they're all going to result eventually in glucose. So we can also say disaccharides, what's their function? Energy as well, because they result in monosaccharides. Now we're ready for polysaccharides, which is, again, a chain of three or more monosaccharides. Usually it's a large chain, like hundreds to thousands of monosaccharides in a row. We are going to cover three polysaccharides, those being starch, glycogen, and then finally cellulose. And it's really important that you realize that all three of these really long chains, all three are composed of just glucose alone. So starch, just a really long chain of glucose, 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 glucose. Same thing for glycogen. Same thing for cellulose. So then you might be asking yourself, why aren't they all the same molecule if they're made up of the same thing? So, what makes starch different from cellulose? Well, I'll talk about that a little bit. It has to do with their structure at the chemical level. So the first polysaccharide that we're going to talk about is called starch. And the first thing I want to mention is starch's biological function. Its function is a storage molecule, a way to store excess glucose in plants. So plants, they do photosynthesis. They produce a tremendous amount of food using sunlight and water and CO2. And, if it's sunny out and they have water, they're just pumping out tons of glucose. And oftentimes it's more glucose than they currently need at that point in time. So they can store a lot of that excess glucose in various places across the plant. Sometimes in the leaf, many times underground, like for instance a potato tuber. Oftentimes they give it as a nutrient to their embryo, so it goes into a seed. Yeah, there's various ways or places to store it. But think of glucose, or think of starch, is just a way to store all this excess glucose in a long chain. Now, in terms of nutrition, major sources of starch are basically the seeds of certain, what we call dry fruits, in other words, grains. So the seeds of wheat, rice, barley, oats, also corn, potatoes, obviously, beans. These are the major sources of starch. Now, that first function was how a plant uses the starch, but then we must also take into consideration that many animals eat those plants and in turn eat their starch, right? And then use that starch for energy. So if you as an animal eat starch from a plant, what does that do for you? Well, luckily, as an animal, you have an enzyme that allows you to take that long chain of starch and chop each individual glucose off so that your small intestine can absorb all those glucose molecules and send them to your own cells to generate energy. So what I'm saying is when you eat starch, you turn it into glucose. You turn it into energy. Interestingly, this digestion process of starch begins in your mouth. So for instance, if you take a cracker or a piece of bread and you kind of just like chew on it for 30 seconds to a minute and you kind of just mash it around in your mouth, try it. It's going to start to taste sweet because you have a special enzyme in your saliva called amylase, which does just that and chops up starch into glucose, and you'll actually start perceiving and sensing the sweet glucose molecules that you are digesting in your mouth. And then I know there's asterisks here, but I just want to mention this. This kind of relates to nutrition. There are two different forms of starch. Now, different foods have different proportions of these two types of starch. And nutritionally, the form of starch that is more healthy for you is this one on top. It's called amylose. And it's a straight, long chain of starch. It's healthier because it takes longer to digest. Because you have to digest it one by one. Chop one off here, kind of like a little Pac-Man, moving your way down the molecule. You go relatively slow. So you slowly increase your blood glucose levels, and it leaves you, in terms of your hunger, more satisfied, more satiated for longer periods of time. Whereas the other form of starch, amylopectin, is more branched. So you can start digesting it from this branch and at the same time from here and from the other end, and you could work your way through the starch molecule quicker. So it causes a greater spike in blood sugar levels, and you're going to burn that glucose really quickly. So you'll be hungry again pretty rapidly. So it leaves you more likely to eat more frequently. So different foods have different proportions of these types of starch. Usually, like an example would be whole breads, like whole wheat, for instance, versus white bread. Whole, it's going to have more of the unbranched, whereas the refined bread is going to be more of the bad kind, the easily digestible version. Same thing with brown rice versus white rice. Brown is going to be healthier, white, not as much. So just a little nutrition side note. Polysaccharide number 2 is called glycogen. Now, it is also just an extremely long chain of glucose, but its function is different. It is the way that animals store their excess glucose. So like I was saying, if you have, let's say you eat 2 whole candy bars within a minute or so, and you have a tremendous spike in blood glucose levels, that's too much to handle, right? So you're going to store some of that in the short term. usually in your muscle cells and in your liver. So how do you store that glucose so you can use it at a later point in time? Well, you store it as this long polysaccharide called glycogen. And then if your blood sugars levels drop, you can then go to that glycogen and break off these glucoses and then break down those glucose molecules. So it's a storage form of glucose. Structurally, pretty similar as starch. Only difference is it's a little more branched. Our last polysaccharide is called cellulose, and functionally, it is very different from starch and glycogen, even though it is also just a long chain of glucose. Now, I'm trying to avoid too much organic chemistry, but in order to understand why the structure of cellulose is different from that of starch, like think about this. A t-shirt is made up of cotton fibers, which are largely cellulose. Pretty tough, pretty strong, you know? My desk here, wood, made up of cellulose and other strong molecules. But it's made of glucose. So what makes it so rigid, so fibrous, so tough? Well, there are two different types of glucose molecules. There's one version of glucose that is flipped upright. It's called alpha glucose. Alpha is down. And there's another version of glucose called beta glucose, which is facing upwards. Now, starch and glycogen are solely composed of alpha glucose, which causes the molecules to take on a granule, kind of mushy form. But cellulose is made up of the other form of glucose, beta glucose. And because of that, it causes the parallel adjacent glucose molecules in the chain to be really linear. And it just lies really flat like a wire, like a fiber. And as you can see here, when you have multiple chains of cellulose on top of one another, the molecules orient themselves in a particular way where you get these chains forming hydrogen bonds perpendicular to the chains themselves. So they're like cross-linked and fortified with these hydrogen bonds. So main thing is it results in a really strong, fibrous, tough molecule. And that relates to its function, which is structural support in plant cells. So one unique feature of a plant cell that animal cells don't have is something called a cell wall, which sits outside their cell membrane, and it's really tough, it's really rigid. Why? Because it's composed of cellulose. Helps the plant with defense and standing upright its entire life. In terms of nutrition, you'll find this in green areas of the plant, so leaves, also fruits, especially like the skin of fruits, many vegetables as well, and whole grains when you don't remove these fibers. So oftentimes cellulose has another name in nutrition, a common name, and that's an insoluble fiber, meaning you can't digest it. It goes straight through your system. It's good for you though. Which brings me to my next point about cellulose. So the first two polysaccharides that we talked about, starch and glycogen, I told you, you as an animal have enzymes that allow you to break down this chain into glucose, right? Little pieces of glucose. But for cellulose, you do not have that enzyme to break these bonds. If you did have that enzyme, you could eat grass and it would do the same for you as eating a potato, you know? It would turn into glucose, but you don't. But you're probably wondering, well, some organisms do eat grass, some organisms do eat wood, and they get energy from it. So, like for instance, a cow and other grazing animals, they can eat cellulose-rich grass, right, a leaf, and somehow that provides energy for them. So what's going on there? Does the cow have this unique enzyme to digest cellulose, which I don't have? The answer is yes and no. The cow itself does not have the enzyme, but in the digestive tract, so the intestine of the cow, they have symbiotic bacteria that live there that do have this enzyme. And the bacteria help the cow out by digesting the cellulose into glucose, which the cow can then absorb for energy. But for you, and most animals that aren't grazing animals, the cellulose will go straight through your digestive tract. You won't get any energy from lettuce or grass. Same thing with termites. They also have bacteria in their guts that help them digest the cellulose in wood. Then lastly, I have a fourth polysaccharide that I want to mention. It's called chitin. It is the primary structural component of two things: the cell walls, which surround all fungal cells, and also it's a component of the exoskeletons of arthropods, which includes all insects, crustaceans, arachnids. Okay, huge group of animals. It's the most dominant in terms of their sheer numbers group of animals on the planet. So for those animals, it has also a structural function. Chitin does. We are now ready for our next class of biomolecules. In that class, this is the lipids. Now, if you remember back to the part 1 video, this is the one biomolecule class that is not constructed as polymers. So there is no monomer name for this class, and there is no polymer name. They are, though, large, complex molecules, but they don't have smaller repeating parts. And there are two major types of lipids here that we're going to discuss, those being fats. We'll talk about two different types of fats: saturated fats, unsaturated fats, and also phospholipids. One thing that does tie together all of the molecules that fall into this class, they all fear water. They're all hydrophobic. And the reason that they're hydrophobic is because if you look at their chemical structure, they're composed almost completely of non-polar covalent bonds. And if you remember back to chapter 2, that means that all of these bonds involve atoms that are sharing pairs of electrons equally. So there are no partial electrical charges in the atoms, meaning they can't stick. They're not attracted to anything that is charged. So they're not attracted to water. It's not that they fear water so much, it's just that they don't really care for water. They don't stick to it. So water likes itself more than it likes lipids. So that's why if you mix, for instance, let's say olive oil and water, and let's say a test tube, you'll have two layers. You'll have the water layer, and then you'll have the olive oil, right, the lipid, fat layer. And it's not so much that they don't like each other, it's just the water likes itself because water is charged. And the lipids could kind of care less. They don't stick to water. So that's why that happens. So the first type of lipid that we're going to talk about is called a fat. The more technical term for a fat is a triglyceride. And this is a, pretty chemically complex molecule. So for our class, I'm going to reduce this complex structure to what I call a cartoon model of it. But before we get to that cartoon model, I do want to introduce the real structure. And the real structure, as you can see here, a lot of atoms, right? Huge molecule. But it has two basic parts to it. The part in gray, and then the three parts in orange. So before I say any more, I want to use my drawing here. Let's see, I'm going to put a line down the center to divide these two sections. That's not working. Let's see. Good enough. All right, so we're in the bottom half here. We're talking about fats. So the first type of fat we're going to talk about is something called a saturated fat. These are the fats that are largely solids at room temperature. So animal fats, butter, things like that. And if we go back to a fat, the reason it has the name triglyceride, the tri part for 3, is because it has 3 of what we call fatty acid, sometimes called fatty acid chains, that are hanging off of the molecule. There's 1, 2, 3. So the orange parts of the molecule are called the fatty acid chains. All 3 are attached to this gray, which we call a backbone of the molecule, which is referred to as glycerol. So those are the 2 parts of a fat. There's the glycerol backbone in gray, and then the 3 long fatty acid chains. And think about what I said in the last slide. This is a molecule that is almost completely composed of non-polar covalent bonds. And that should be really apparent when you're looking at the 3 fatty acid chains. Carbon to carbon or carbon to hydrogen. Those are all non-polar bonds and responsible for its hydrophobic nature. So going back to my drawing, first thing I'm going to do is label it. So this top part is the gray part from the previous picture. It's called glycerol. That's the backbone. Pretty straightforward. It's 3 carbons. 1, 2, 3. And then completely surrounded by hydrogens. And then at the bottom, each carbon is attached to one of the 3 fatty acid chains. So since the 3 fatty acid chains are pretty much the same, I'm just going to focus on one of them and just say, well, it's the same for the other 2 without drawing it 3 times. So we're just going to focus on one chain, even though a fat really has 1, 2, 3 of these going on at the same time. So this is what it looks like. You basically have what was once a carboxyl group here. But if that part makes you uncomfortable, kind of ignore it. I want you to focus on starting down here, past that. So if you skip past this carboxyl group, then you just have carbon, 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 and it goes on for a while. On average, each fatty acid chain of a fat is anywhere from 16 to 18 carbons in length. Now there's all different types of fats and they have different names, like if you look at your food, like oleic acid or stearic acid or palmitic acid. Those names depend on the number of carbons and therefore their length. And obviously we don't want to watch me draw out 18 carbons in a row, so I stopped at 1, 2, 3. I just put ... This, goes on until sometimes I'll just put it right here. 16 to 18 more carbons. And only part I left out is that these carbons, remember, each carbon has to be involved in 4 bonds. If we, for instance, look at this carbon right here, how many bonds is it involved in currently? Well, the answer is 2. 1 here, 1 here. But it needs 2 more to equal 4. So that's where all these hydrogens come into play. So if I finish this, it looks like this. That's where the name comes in. I'm kind of running out of space here. I'm putting an H right there. A little messy. But it's completely saturated. It's completely surrounded by the maximum number of hydrogen atoms. Okay, it's a saturated fat. That's going to be a little bit different when we talk about unsaturated fats, because there'll be a slightly smaller number of hydrogens and it won't be completely saturated. We'll talk about why that is in a moment. Last thing I want to do here is, or second to last thing, I want to label this whole section. So this is a fatty acid chain. And remember, if we were to complete this diagram, a fat has 3 of these. So it would have a second one here and a third one here. And then now we want to convert this into our simplified cartoon diagram. And in my simplified model, this rectangular black box is going to represent glycerol. And then lastly, we will depict the 3 fatty acid chains with just lines. So 1, 2, 3. So that's our saturated fat. And I'll put a label. So there we go. And again, this glycerol just represents this. The 3 carbons at the top. So that's the structure. In terms of their function, you probably already know this, but fats also supply energy, just like carbohydrates. In terms of how much energy they supply, in general, it's twice as much per gram when compared to a carbohydrate. And they're better, fats are, for the long-term storage of energy, which can then be used way later on in stressful circumstances, or starvation, or hibernation, or extreme environmental conditions. Okay, it's kind of like your last resort, or it can be, for energy. And it's really good to store because of its hydrophobic nature. So you might ask, well, why don't I just store all my energy molecules as carbs? Well, you would weigh like 4 times as much, because you'd have all this water globbed onto the carbohydrates because they like water, and it would be really energetically inefficient. Whereas you can store fats and not weigh as much because it doesn't collect all this water with it. But they do release more energy when broken down within a cell than a carbohydrate. So I recommend watching this video, really quick, nice, simple, on the difference between saturated fats and unsaturated fats. But even if you don't watch that, I'm still going to take you through the basic differences right now. And As I said, saturated fats are generally solids at room temperature, and that is because the fat molecules are really flat. Okay, it's linear. So if you have many fats, saturated fats together, they all pack really closely to one another like a stack of papers. And they don't move as freely at higher temperatures, so it takes a lot of heat, a lot of energy, a lot of temperature increase to get them to transition into the liquid state. So that's why they're generally solids. And structurally, what distinguishes a saturated fat from an unsaturated fat is that saturated fats, if you are to look at their fatty acid chains, there are no what we call double bonds present. So all of the bonds that you will see between adjacent carbon atoms, they're always just a single covalent bond. But that will not be the case when we are talking about unsaturated fats, which leads me to unsaturated fats. So here we go at the bottom. Let me draw my line. Overall, structurally, very similar to a fat. It's got glycerol at the top. It's got 3 fatty acid chains. I'm only going to draw one of them for time's sake. But the key structural difference is that an unsaturated fat has one thing different from a saturated fat, and that is the presence of at least one double bond, as you can see here, between adjacent carbon atoms in at least one of the 3 fatty acid chains. So it's long, right? We said it goes all the way down to 18, and there's 3 of them that go all the way down to 18 carbons long. If you just have one single double bond between any of your 2 carbons, this fat is automatically an unsaturated fat, and it's automatically going to be a liquid, in other words, an oil at room temperature. Okay, so that is the key distinguishing feature. Unsaturated fats have one or more double bonds in their fatty acid chains. Now, some unsaturated fats can have more than one. We call those polyunsaturated fats. Generally, what you would consider the good fats in terms of health, nutrition, like those found in many seeds of plants and also fish. So like your omega unsaturated fats. And then if you're a fat that only has one double bond, you're referred to as a monounsaturated fat. Like for instance, that which you'd find in avocados or something like that. So why does it get the name unsaturated? Well, let's kind of finish this drawing here. Remember, each carbon needs 4 bonds coming off of it. How many does this carbon have currently? Well, your answer should be 3. 1, 2, 3, but it needs one more bond. Same thing with this carbon below it. It has 3 bonds, so it needs one more. But all the other carbons that are not involved in the double bonds are going to have 2 hydrogens coming off of them. Now, compare that to our previous drawing. Do you see how there's more hydrogens surrounding this molecule when there's no double bond? Each one has 2, right? 1, 2, 3, 4, 5, 6, all the way down. An unsaturated fat is not completely saturated with hydrogens. It's not completely surrounded by hydrogens. There's less. There's only 1, 2, right? So like 2 less. So that's where it gets the name unsaturated. It's not completely surrounded by hydrogens. And that does something to the overall shape of this molecule. It creates a kink or a bend in the fatty acid tail that has the double bond. So if we look here, this is what I'm talking about. So there's your double bond right there, and the rest of the tail bends. And that's why this is a liquid at room temperature, because if you try to stack multiple unsaturated fats together, all these kinks and bends make them not well packed together, so they move more freely at room temperature, resulting in their liquid state. And that is why we're going to give the cartoon structure of an unsaturated fat a little bit of a different depiction. So we want to recognize that bend because of the double bond. So we're going to draw one of the fatty acid tails with a bend in it. Like that. So that's my cartoon diagram for an unsaturated fat. Compare that to a saturated fat. Here they're all straight, flat, because it's saturated, right? It's linear. Here's your unsaturated fat. The last type of lipid I'll talk about in great depth is the most important for our class moving forward. And this type of lipid has an important structural function in all living things, in all cells, because it is the primary component of membranes. And as we will learn in chapter 4, one feature common to all cells, whether you're a plant cell, an animal cell, a bacterial cell, doesn't matter. You have to have a membrane. You have to have a barrier outside the cell on the perimeter that says, well, everything inside of this is the cell, right? Everything outside is the environment, everything else in the world. So that's a common feature of life. And what I'm saying here is these membranes, this thin barrier on the edge of life that defines life, really. This barrier, this membrane itself is composed of a special type of lipid called a phospholipid. So I'm not going to go too in depth into its structure here because we're going to talk about it again at the start of chapter 5. But I do want to point out, number 1, it has a structural role. It is the primary component of all cell membranes, and also the membranes that line and define all of the organelles within eukaryotic cells, whether it be the nucleus, the mitochondria, the lysosome. Every organelle is surrounded by a membrane composed of phospholipids. Next, we want to mention that this is, as I said just a bit earlier, a unique molecule. And it's unique in the way that it interacts with water. Unique in the sense that half of the molecule likes water, half of the molecule fears water. So half of it's hydrophilic, the other side of it's hydrophobic. And that creates a really unique structure called a phospholipid bilayer, which is a membrane, which we'll talk about in chapter 5. We have a very technical term for this idea of both liking and disliking water at the same time. It's called amphipathic. But for here in chapter 3, I just want to go over the basic structure, and most importantly, the cartoon structure of a phospholipid. Now, for the fats, I kind of made that up for our class. I think it should be commonly used. It makes sense. But the cartoon version of phospholipids is not just my gentle way of teaching fats. It is commonly used in biology, and it's the way that we draw phospholipids. And I'll draw it many, many times moving forward in this class. So let's start with the real structure and then work our way to the cartoon. So the real structure, kind of makes one cringe a little bit. A lot of chemistry, a lot of organic chemistry. But it has some similarities to fats. And those similarities are, it has a glycerol backbone with 3 carbons. It has some fatty acid chains coming off of it. it's not like a triglyceride. It's not like a fat. It only has 1, 2, right? So it only has 2 fatty acid chains. And also it has something unique going on at the top of the glycerol. It has one of those 4 functional groups that we learned about in the part 1 video. And if you recognize that, that is a phosphate group. And it's that group right there that is responsible for the water-loving nature of a phospholipid. So more importantly for us, let's transition into the cartoon diagram. So the way we draw this is, I don't want to use bold, so let's change that up. We draw a circle at the top. We have a name for it. This is called the head region. This is where the phosphate group is. This is the part of the phospholipid that loves water. Then, coming off of it, the conventional way to draw the tails. Now, we draw them kind of like squiggly. That doesn't necessarily denote that they're both unsaturated, although one of the 2 tails is. But the conventional way to draw this out looks like this. I'm going to use a different color to distinguish it from a fat. Like that. Okay. I feel like those are kind of ugly. I'm going to redo that. Something like this. There we go. It kind of looks the same. Oh well. So those 2 fatty acid tails, let me put a little bracket here. Oh, those are part of the tail region. Remember, we said we sometimes call the fatty acid chains fatty acid tails. Those, they mean the same. So this is the tail region, and this is the region that does not like water. Because if you look at a tail, right, of a fat or a phospholipid, it's a bunch of non-polar bonds. All right, so again, really important because we're going to expand on that model in chapter 5. I'm going to draw it many, many times. So definitely know that that's a phospholipid. The easy way to tell it from a fat is that it has 2 fatty acid tails as opposed to 3. Then here I just want to mention, fats and phospholipids are not the only lipids out there. They're the ones that we here are going to focus on, but other examples of lipids include waxes. For instance, the wax that coats a leaf that holds in water. Many vitamins, many that you are familiar with, like A, D, E, and K. And then also steroids. Cholesterol, which is an example of a steroid, is found within all of our membranes that helps maintain their consistency between, or keeps them liquidy and helps us make sure they don't solidify at cold temperatures. But also other steroids, like for instance, estrogen or testosterone, things like that, that act as hormones in our body. Many of these are lipids. Now, it doesn't look a lot like a fat or a phospholipid, because remember, this group is not made up of small parts put together in the same way. It's just one big group. And the only thing that ties together this group is that all of these molecules generally don't like water, with the exception of the head region of phospholipids. All right, now we're ready for our 4th and final class of biomolecules. That class includes it's called the nucleic acids. Now, as a review, the monomer molecules, right, the building blocks of this class are referred to as nucleotides. And another term for a nucleotide is a base. Okay, so those 2 terms mean the same thing. And a little bit easier than, let's say, the protein class. Remember the protein class? They were made up of amino acids, and it was so complicated that I said, don't even worry about the names or the structures or anything about these amino acids because there's 20 of them. It's too much. Well, for this class, it's a bit more manageable, and you do need to know the 4 monomers. Those 4 monomers, which you may have heard of because they make up the genetic code that all life is written in. Okay? Those 4 nucleotides are called guanine, just the letter G for short, cytosine, or sometimes just called C, thymine, T, and lastly, adenine, A. Or we can just say G, C, T, and A. Those are the 4 bases, the 4 nucleotides that all genetic material, all DNA, is composed of. So all of your genes are long sequences of G's, C's, T's, and A's. And this code is universal to all life. So regardless of whether you're an elephant or a chimpanzee or a palm tree, your genes are written in the same molecules, these 4 molecules. What distinguishes a chimpanzee from a human from a palm tree is the unique sequence of your genes, the particular order of the G's, C's, T's, and A's. Because as we're going to learn, these 4 bases, their function is largely to hold, store, and transmit information. Okay, it's kind of like a computer or a library in the cell. It's a blueprint for building all of the proteins that, for instance, make you a human being or make a palm tree a palm tree. Okay, it's the information needed to build all of the molecules that make you the species that you are, responsible for your structure, your particular anatomy, your physiology, your enzymes. Everything about you is due to the information that you inherited from your human parents. And that information was DNA. 23 chromosomes from your mother, 23 from your father. So this is a very cool class. It's really kind of the core of biology is DNA. Okay, you can't escape it. It's the foundation of pretty much most chapters and most units moving forward. So really important information here. But first, we're going to keep the theme consistent with the previous classes of biomolecules. Here in Chapter 3, it's very introductory. We just want to expose ourselves to the basic structure and function of both the monomer and the polymers. So here we're starting with the monomers, nucleotides. This is one of those important molecules that you should be able to recognize. Okay? And I want you to realize, you should know, that a nucleotide has 3 basic parts to it. Number 1, it has a phosphate group. Called ribose. And then up here it has a really complex structure called the nitrogenous base. And it's this part here that's slightly different from A to T to C to G that distinguishes one nucleotide from the other 4, or the other 3. Now, as with the other 2 classes, right, carbs and proteins that have polymers, same thing for this class. If you put, say, 3 or more nucleotides together into a chain, it then becomes a nucleic acid. Okay, so nucleic acid is a chain of nucleotides. There are just 2 types of nucleic acids, both very important. First, there's DNA, which stands for deoxyribonucleic acid. And then there's RNA, which stands for ribonucleic acid. We're going to talk about both of these heavily in this class. We're primarily going to stick with DNA here for our introduction. And I want you to realize, not only is DNA a long chain, it's 2 long chains running what we call anti-parallel to each other. It's a double-stranded molecule. So here's one chain in purple, and then it has a sister chain. It's a double-stranded molecule. And if you were to look at these two strands, like look here, when the bases, the nucleotides, are adjacent to one another from each strand, they pair together in a specific way. And this is very important. We call it the complementary base pairing rules. So what I'm saying is if you look at one strand of DNA, and let's say you see a T, that means on the other side, the other sister strand, you're always going to see an A. So there's rules to how they're situated across from one another, the way that they pair up. And those rules are A always pairs with T, and C always pairs with G. So you got to remember that, A to T, C to G. And if you want to get into the chemistry of it, those two adjacent nucleotides are actually sticking together via hydrogen bonds. And it's the collective force of all of these hydrogen bonds along the whole DNA molecule that holds the two strands together. So they stick. And since this is so important, I want to take the time to do a practice problem with you. All right. So the problem says, let's pretend that we are observing the nucleotide sequence of just a single strand of a DNA molecule. I'm giving you that sequence here in blue. We're saying the sequence is A, C, G, T, T, and so on. Using what you're given here, what is the sequence, the nucleotide sequence, of the corresponding strand? To answer this question, you need to know your base pairing rules. So you must know that A pairs with T and C pairs with G. So I'm going to work through this with you. I'll use a different color for the other strand. We're just going to write the sequence of the other strand just below the top strand. So when we see, let's put the rules over here. So A to T, C to G. So if you see an A, well, across from it must be a T. If you see a C, across from it must be a G. So I won't say that every time. I'll finish it here. But definitely do this on your own. Maybe make up your own practice problem. That's it. So just understanding the basics of the base pairing rules. Next, and really very important, we'll spend an entire chapter diving deeper into the basic functions of DNA, which makes this chapter, really chapter 3, so tough because there is a lot to say, but it's still introductory. We're still just grazing over things. So what I've done here is I've basically summarized an entire field of biology, or for our case, entire chapter 10, into a single slide. Okay? And these are the 3 major functions of DNA. And these are common not just to animals, not just to humans, to any living thing. This is how every cell uses its DNA. And the most important function is using DNA as genetic information. So using it as information to store, using it to store the information for building the various proteins in your species, in your body. So remember, we said there's all different types of proteins. You got all these enzymes, you have all these membrane transport proteins, you have hormones, you have receptors, you have movement proteins. How did you just magically know how to build all that from scratch? No, you started as one cell, a fertilized egg. From a unification of a sperm and an egg, you became one whole cell with both the 23 chromosomes from your mom and the 23 from your dad. Now, within that single cell that you started as, in the nucleus of that cell, was all the genetic information that you need to build your entire body. So across those 46 chromosomes that you had, and that you still currently have in all of your cells, are tens of thousands of different what we call genes. And each gene is the recipe or the blueprint to build a specific protein in your body. So no, you had to inherit that information from your parents. And, that's how genetic diseases work. If you received a faulty, mutated version somehow, then you may not have the perfect ability to build all of your proteins. You might be missing something. But ideally, you want to inherit, 23 chromosomes from each parent without any mutations, so that you have the ability to build all of your essential proteins. So in a simple sense, what am I saying? I'm going to write this out. Now the point is, I don't expect my written description here to be like a dictionary definition, because I'm doing it on the spot. I'm not looking at I don't have any notes. It's coming straight from me. I kind of want it to be freestyle. So let's paraphrase what we had just read. So what is the primary function of DNA? The primary function is to store, to store information. That's the key word. And we'll learn a bit more about how we store this information moving forward in the class. They're stored as little segments of DNA called genes. And as we're going to learn, each gene holds the information for building a particular type of protein. Okay, so I'm going to finish this. To store information as genes for building polypeptides. And basically each gene that you have is the information to build a unique polypeptide. So one gene, one polypeptide. As a human, if you're interested, you have, over 20,000 different genes. And that's it. I'm satisfied with that basic description. So it's genetic information. Then we'll get to number 2. So let's go back. Now I want to say one more thing about the first function. These polypeptides, remember, proteins are so important. They do everything. They're responsible for all of your functions, all of your structure. So we can therefore say that these genes, this DNA that codes for those proteins, right, that DNA programs and holds the information for all of the structures and functions of your entire body and each and every one of your cells. Then we can also say DNA is also considered a hereditary molecule. It's the molecule of inheritance. It is the molecule that a parent physically passes on to their offspring, right? You don't give your child a nose. You don't give your child hair. You don't give them blue eyes. You don't give them proteins. You don't give them carbohydrates. You don't give them fats. What do you give them in a sperm and an egg? You give them DNA. You give them genetic material. You give them the instructions for building the same kind of nose you have, the same color eyes, okay? So you give them information, which is used to build physical proteins, okay? So we are just going to say DNA is a very special molecule because it is the molecule of inheritance. It is the molecule that is physically passed on from parent to offspring. It's transmitted from generation to generation. So you are a genetic composite of all of your ancestors going back, well, as far back as you want to go in time. Transmitted from So I'll tell you the truth. I intended to have my laptop open for this part so I could look at my, I wrote it anyway, so it's not like I'm reading something from a book, but I forgot my laptop where I'm at, so I'm doing this from my head. I think it's turning out all right. It looks the same as it does in my slides, but transmitted from parent to offspring. Not that I need the slides, I just want to make it consistent. That should do it. Yep. So also realize genetic and hereditary are kind of mean the same thing. It means it's passed from parent to offspring. And then lastly, we'll also cover this last point in this class. DNA is also pretty unique as a molecule because it has the information in it to replicate itself. Think about it. I just told you that all of us, well, pretty much every animal, yes, every animal, starts as one cell, a fertilized egg. Yet you've grown up into this huge multicellular organism composed of trillions of cells. And each of those trillions of cells are descendants of that single fertilized egg that you started as. So you went from 1 cell to 2 cell to 4 to 8 to 16 to 32, all the way to a couple trillion. Right? And I told you that that original first cell that was you, the fertilized egg, had in it all of your genetic material, 23 chromosomes from mom and 23 from dad. Well, how did you make sure that that information got passed on to, well, all of your other cells? Because now you're a trillion cells, and hopefully, right, all of those trillions of cells also have the 23 chromosomes from your mom and the 23 from your dad. So what I'm saying is, when you grow and develop from 1 to 2 to trillions of cells, each time a cell divides in your body by something called mitosis, you need to make sure that those 23 chromosomes from mom and 23 from dad are faithfully copied and replicated, so you can give a copy of all of your genes to each new cell. Okay? That's a very important component of life, the ability to build and make copies of your DNA. So DNA has the ability to replicate itself. And this is something else that we will cover in greater detail in Chapter 10. All right. And then a last little word here about monomers and polymers in general. Maybe you've realized this, but one thing that happens when you have these long polymer chains, you have great potential for building many different types of chains that vary not in the monomers that they're made of, but in the unique sequence that you put them in. So think about DNA. Think of how many life forms there are currently on the planet. Millions. Think of how many that have ever lived on the planet. Many, many that are currently extinct. And all these different organisms had different genes. But all of these genes are made up of A, G, C's, and T's. So how do you get this great diversity in DNA and genes when you only have 4 building blocks? Well, the key is the sequence that you put them in. So think of it this way. It's kind of, there's some math to it. If you have a segment of DNA that is, let's say, 1,000 nucleotides long, that's about a general length of a gene. how many different sequences of those 4 nucleotides could you have? Well, the answer would be 4 to the 1,000th power. That's 4 times 4 times 4 times 4, 1,000 times. That's so many different combinations of A, G, C's, and T's that the final answer is almost infinite. So the key to having such great, diversity in life in terms of structure and function is due to this concept of, we're all made up of the same pieces, the same, glucose, glycerol, fatty acids, phospholipids, A, T, C, and G, 20 amino acids. That's pretty much life. But you can put them together in so many different orders when you construct them into chains that the types of polymers you can build is almost endless. And that's why you have crazy differences like gills and feathers and scales and skin. And these are different proteins that arise from different polymer sequences. So, kind of interesting. And we want to end this by expanding on, as I said, the most important of the 4 classes of biomolecules, especially for us moving forward. We're going to talk a little bit more about DNA. And we're going to focus on what I call the various structural levels of DNA. And the way I like to do this, even though I have it written out 1, 2, 3, and 4, going from small to large, I'm going to flip it around. I'm going to start really big and work our way down to really small. So I'm actually going to go backwards from 6 to 1. And I'm going to use a picture as I take you through these structural levels. This is another section that I really encourage you to comprehend it well now. Because if you don't, it's going to bite you twice. Because we're going to cover these same concepts multiple times. So you might as well get it now and then not have to relearn it a second time. So, important section. All right, so we're going to start with DNA terms, starting with the biggest structures. Now, again, I'm going to use my picture. Here's my picture. So what do we have going on here? We're pretending that we're looking into a eukaryotic cell. Let's say it's a newly discovered organism. It doesn't even have a name. We don't know what it is. And let's say, it's not a human, so it doesn't have 46 chromosomes, but this newly discovered organism happens to have, how many are there? Let's count. 1, 2, 3, 4, 5, 6. I could have picked anything. I guess the main thing is I don't want to draw 46 chromosomes. That would be nuts. So we're going to say that this made-up example here, it's an organism that has 6 chromosomes, right? And we're going to use its 6 chromosomes to take us through the various structural levels of DNA. And we can apply these to humans, okay, as well. So there we go. Now the biggest term that we're going to start out with is something called, I'll keep the numbers consistent, so number 6. It's called your genome. So what is that? That is all of your genetic material that you inherited from your 2 parents. So if we're talking humans, that means all of the nucleotides across all 46 of your chromosomes. As a human, that's a lot of information. How much? Well, 6 billion nucleotides. So 6 billion A, G, C's, and T's. We all have that. That's our genome. For this simple diagram here, the genome would be basically let me fix that. I think this will work. Not bad. What I'm saying is, not working out. I think you get what I'm trying to do. So I'm going to use my own ugly line to get to this one here. There we go. Let's see if I can fit one more in here. Not really, so I'm gonna have to do my own line. There we go. So what I'm trying to say is the genome is all the genetic material collectively put together. So if you're a human, it's all 46 of your chromosomes. If you're this made up organism here, it's all 6 of its chromosomes. Okay? All of the DNA in the nucleus of each and every one of your cells is your genome. So we will jump back to the definition of a genome. It's the organism's complete set of DNA. You have one copy of your genome inside each of your trillion plus cells. So what I'm saying is, in every one of your cells, your skin cells, your heart cells, your muscle cells, your liver cells, each one of your trillion plus cells has a nucleus inside. And inside each nucleus is the same thing. The same 23 chromosomes from your mom and the same 23 from your dad. 6 billion nucleotides of information. That's insane. That's a lot. How insane is that? Well, we know how small molecules are, right? If you took the 46 chromosomes from just one of your cells and stretched it all out, it would be, they say, several meters long. Okay? Like the length of your body long. That's wild, right? A lot of information. Next, getting a little bit smaller. If you look at a chromosome, or any part of DNA, you'll notice that the DNA itself is coiled and wound upon tiny little spherical proteins. And this helps to pack in all of this information into a really tiny space. Right? That's a lot. Meters of information inside a tiny cell. You have to coil it like a spool of yarn and pack it into the nucleus so it fits. We do this by packing our DNA and winding it around proteins called histone proteins. So I'm only going to do it for one of the DNA molecules. The blue dots represent the proteins that the DNA is wound upon. Now, this next term, which I'm getting to here, called chromatin, refers to your DNA, right, the nucleic acid, and the histone proteins that it is wound upon. So if you take into account both the protein and the DNA wound around it, we call that chromatin. Getting smaller. So chromatin is basically kind of the same as your genome, actually. It just takes into account the proteins that it's wound upon as well. So it's kind of just a different term, not necessarily smaller than a genome, kind of the same level. But the next term is smaller. That is a chromosome. Now, as a human, in each of your cells, you have 46 different chromosomes. 23 from your mom, 23 from your dad. So I can number the chromosomes here. There's no particular order to it. We have 6. And the way I've drawn these is kind of a classical way to draw a chromosome, which is in the duplicated form. This probably looks less like a chromosome if I were to draw just like half of it, right? It's still a chromosome, but I like to draw it like this because this is the more recognized version. Realize when we draw it out like the way that I have, these two molecules are replicas of one another. They are the same thing. It's been duplicated. Okay? So we have 6 of them. More importantly, each chromosome is one DNA molecule. It's one extremely long, double-stranded DNA molecule. Well, just about how long? Well, with respect to humans, on average, 50 to 300 million nucleotides in a row. That's pretty long. So that's a chromosome. Each chromosome has hundreds to thousands of different genes scattered upon it. But the whole thing is not just genes. Okay, most of it is not genes. Genes are every once in a while a little blueprint, little recipe to build a polypeptide. So let's label that. For one of them, we can say this whole thing here. What is it called? That's a chromosome. And it's one extremely long polymer of DNA. And going back to my slides, that was number 4. Getting smaller. As I just said, along each chromosome, there are many genes. And a gene is a particular arbitrary stretch or segment along a chromosome that is the information, very important, for building a single type of polypeptide, which, if folded correctly, and once it assumes its proper 3D structure, becomes one of your proteins, right? So on average, a gene is anywhere from 10,000 to 15,000 nucleotides long. So let's draw that out. I'm going to pick, let's go to a different chromosome because we have a lot going on number 1. Let's go to number 6. And I'm going to say, just making this up, let's pick a different color. I'm going to say from this X here, moving to the right, to that X, that's a gene. Okay, so this stretch right here, we're going to say that little bit is a gene. And each chromosome has several genes, so there might be another gene that goes from point A to point B, maybe another one from here to here, another one from here to here. Gene, gene. Okay, so each chromosome has various genes. So I'm just going to focus on one, and it's between the X's. There we go. I'm going to label that. And it's kind of confusing because I numbered the chromosomes. I'm going to get rid of that because it's confusing with my other numbers I have going on. So we are now, with respect to the slides, at number 3, working our way from large to small. I'm going to draw out a gene up here as well. And basically I'm taking this point here, okay, I'm going to circle it, that gene, that segment between the X's, and I'm going to zoom in on it here. Okay, so we're going over here, we're going to zoom in on it. And again, a gene's like 10,000 to 15,000 nucleotides long. Obviously you don't want to watch me write out 10,000 A, T, C's, and G's. So we're going to pretend it's 7 nucleotides long for simplicity. We're also going to practice our base pairing rules at the same time. So I'm giving you the sequence of one strand. What would the other look like? Like this. And again, it keeps going because it's longer than that in reality. And I'm going to put a yellow line here at the bottom. Maybe it's not yellow. Does it really matter? Looks more like that. Oops. That represents our gene. All right. It's a certain stretch of DNA along a chromosome that codes for a polypeptide, codes for building a polypeptide. Then the remaining two structures are not on my picture. We've already covered them. DNA, right, that's our polymer, a specific type, right? There's DNA and RNA. DNA is a polymer, and it's a long segment of nucleotides put together in a chain, and it's also double-stranded, and the two strands are complementary to one another. And then our smallest structure, which we are already familiar with, is the monomer for this class, a single nucleotide, also known as a base. And to keep my video here shorter, I'm going to say watch this video on DNA on your own time, please. Here's just another visual illustration of these structural levels of DNA. Little recap. All of this together would be the genome. If you were to take into account the DNA plus the proteins that they're wound upon here, that would be your chromatin. If you were to just pick one of these structures, one extremely long double-stranded polymer, right? Look how long that is, really long. All coiled up. That's a chromosome. And then not shown, but particular stretches along that chromosome are genes that code for building polypeptides. And I believe that's it for the video. Thanks for listening.